Hey guys, so today's lesson we're going to be talking about enzymes and their effect on different types of reactions. So as we're going through this lesson on enzymes, we're going to be talking about what the difference between the term catalyst and enzyme is. We're going to define things like active site, substrate, and where those actually are on the enzyme or how they relate to an enzyme. We're going to talk about what some of the different digestive enzymes are and what they do, since our next lesson is going to be all about the digestive system. And then we're going to describe different factors that affect rates of reactions involving enzymes and how something called feedback inhibition works. So a catalyst is just essentially a molecule that helps to increase um, the rate of reactions. So in order for reactions to take place, molecules need to not only collide with one another, but collide in the correct orientation. Most reactions actually don't occur on their own or spontaneously, or they would occur very, very slowly because it's difficult for this to happen, for molecules to collide in the correct orientation. You could speed up a reaction by increasing the temperature, since that means molecules are moving around faster and colliding more often, but it's not always safe to just crank up the temperature on your reaction. A safer way is often to use a catalyst, which is just a chemical that's going to increase the reaction rate of the chemical reaction without actually being a part of the reaction. So it's not going to affect what the products of the reaction are, just how quickly you're making those products. Essentially, it makes sure that anytime molecules collide, they're going to collide in the correct orientation. So if I am trying to react these two molecules here, they can collide in a lot of ways that is not going to react. They need to collide in this orientation in order to react and form our product. The catalyst is going to make sure that every time these molecules react with one another, they're colliding in that correct orientation. And by doing that, it's going to decrease something called the activation energy of the reaction. The activation energy of a reaction is how much energy the reaction needs in order to get going. If you have a catalyst that's doing a lot of the work for you, the activation energy of the reaction is going to be lower. Enzymes are essentially just a type of catalyst that deals with chemical reactions taking place inside of the cell. The molecules that the enzyme works on are called the substrates. You can have either multiple substrates like this that are combining to form a product, or you can have a single substrate that is breaking apart to form multiple products. Each substrate is going to have a specific enzyme that it binds to. So you have like hundreds of different enzymes in your body to help with all of the different types of chemical reactions you have going on. Again, the enzymes decrease the activation energy that would be required for the reaction by bringing together the substrates in the correct orientation. Enzymes have something called the active site, which is where the substrate attaches to the enzyme and reacts. And enzymes tend to have names that end with ASE, so you can usually identify them by things like lactase or amylase. They end in that ase ending. So this is an example of what this could look like. So here we have at the bottom here, the gray would be our enzyme, and this lime green would be our substrate molecule. The area at the top of the enzyme here that binds with the substrate is called the active site. And then in this case, you have a single substrate that's being broken apart into two different products. Digestive enzymes are just enzymes that help with the digestive process. So you have carbohydrases, which break down carbohydrates. So that includes things like sucrase, which breaks down sucrose, and amylase, which breaks down starch. You have proteases, which break down proteins into smaller polypeptides, and then peptidases, which break those polypeptides into amino acids. You also have lipases, which break down lipids into their parts of fatty acids and glycerol. There are a bunch of different factors that will affect how well these enzyme reactions actually work. pH is one of them. So different enzymes work best at different pHs. Most enzymes kind of have like a specific ideal pH. And if they get outside of that pH range, the enzyme will essentially denature and stop working. So yeah, pH can alter the structure of the enzyme, so cause it to change its shape or denature. So for example, pepsin is an enzyme in the stomach. It works best at a pH of about like 2. If you were to get it up to more of a neutral pH of like 7, it wouldn't work anymore. But there's other enzymes that work better at higher pHs. So it just depends on the enzyme. Substrate concentration is just essentially how many of the reactant molecules do you have. The more substrate that there is available, the faster the enzymes can catalyze the reactions up until the enzyme becomes a limiting factor. So essentially, if you have a low substrate concentration, the enzyme is always like having to kind of search around and look for a substrate to bind to and then catalyze. However, the more substrate you have around, so the higher the concentration of substrate, the faster it is for an enzyme to bump into a substrate and then just be able to latch on and catalyze it. That will increase, so the increase of substrate concentration will correlate with an increase of the reaction rate up until that enzyme is just like surrounded by substrate and it's just like working as fast as it can. And then at that point, the reaction rate just kind of plateaus. And then temperature, same kind of idea. Reaction rate will increase as temperature increases because the temperature increasing is going to mean that the kinetic energy of the molecules increases. They're moving around faster. They're bumping into the catalyst more often, which means that the catalyst or the enzyme can actually do its job more quickly. And again, that'll happen up until a certain temperature, at which point then the enzymes actually start to denature and the reaction rate will drop off again. So these are 3 graphs that kind of summarize this one. So this first one is just showing you the effective pH. So some enzymes like pepsin will work the fastest at a pH of 2. And as you get outside of that range, it's going to slow down until, in the case of pepsin, if you get up to a pH of like 4, it can't even work anymore. It's denatured too much. And then there's other enzymes, like in this case, trypsin, which works best at a pH of like just about 8. And if you get too far outside of that, the reaction rate drops off. This one up here is showing the effect of substrate concentration. So the higher the substrate concentration, the higher the reaction rate. So you have this increase of reaction rate as the substrate concentration increases up until it just kind of plateaus. That is where the enzyme is just working its absolute fastest. And then with temperature, you also have an increase of reaction rate as the temperature increases up until you hit the point that the enzyme starts to denature, and then it drops off as the enzyme stops functioning. There's also a process called competitive inhibition that can affect how quickly enzymes can work. A competitive inhibitor is when you have a molecule that fits into the active site of an enzyme but is not the substrate. Essentially, it competes, hence competitive inhibitor, it competes with the substrate for access to the active site. So if a competitive inhibitor binds with the active site of the enzyme, that means a substrate can't bind, which means that that enzyme is essentially not functioning at that point. It's out of commission. So yeah, when the competitor attaches to the active site, it inhibits the substrate from being able to bind to the active site. So a competitive inhibitor slows down the reaction rate. Usually metabolic pathways, which are just different reactions that take place inside your cells, usually they occur in a bunch of different steps. Think of like cellular respiration. You have first to break the glucose down to the pyruvate, and then you have to turn the pyruvate into acetyl-CoA, and then you have to go through the Krebs cycle. Like there's all of these steps that have to happen in a certain sequence. And enzymes regulate each of those steps of the sequence. As a product of the chemical reaction builds up, that product can actually go back and interfere with the function of an enzyme from one of the earlier stages. So this would be like, for example, if the buildup of ATP shut off the function of coenzyme A in pyruvate oxidation. So what happens is the product of the chemical reaction is going to go back to one of the previous enzymes, and it's going to bind with something called the regulatory site of the enzyme. So it's not binding to the active site like our competitive inhibitors do. It's binding to a different site called the regulatory site on the enzyme. But when the product binds to that regulatory site, it actually changes the shape of the active site. And once the active site is a different shape, the substrate can't bind to it anymore. So once again, it's essentially putting the enzyme out of commission for a while. This process is called feedback inhibition. Something from the end of the process is going back in a feedback loop to inhibit the chain of chemical reactions by inhibiting one of the enzymes from working. So this interference is going to slow down the reaction rate until that buildup of final product can dissipate, and then the product will pop out of that regulatory site, and this sequence of reactions will pick up again. So essentially, this is what it would look like. So this is our enzyme. This in here is our active site that the substrate would bind to. And down here is a regulatory site. And what happens is the product of one of these reactions binds to the regulatory site. That changes the shape of the active site up here. And now the substrate can no longer bind to the active site. This enzyme is out of commission. So really, these are just like the sequences of all of the different reactions that could be taking place. All of these different enzymes are regulating each of the different steps of this reaction. And if the final product goes back and interferes with the first enzyme by binding to its regulatory site, this entire reaction shuts down until we can get rid of some of that final product. Okay, so that's it for enzymes. Make sure you know all of the different factors that can speed up and slow down enzymes, and specifically how they work. So for example, with catalysts, you want to know that they work by decreasing activation energy and making sure molecules collide in the right orientation, as opposed to say temperature, which increases kinetic energy. Okay, let me know if you have any other questions.